Assalamualaikum and a very good day to everyone. My name is Khairunisa Alia binti Muhammad Rosdi from Group 2. Today, as a part of our Crop Science AGR499 course, I will be presenting on Chapter 1.4, Seed Development. Seeds are the foundational unit of modern agriculture and crop production. Every major grain, legume and fruit crop we rely on for global food security begins its life cycle as a tiny, highly specialist seeds. In this presentation, we will explore the fundamental biological definition of a seed, break down and compare its internal structural autonomy, trace its development pace from fertilization to maturity, and finally, look at the mechanism behind its germination types. This brings us to why studying seed is so essential in crop science. Seeds are important for plant reproduction and crop production. For the plant, it ensures survival across seasons. For us as a crop scientists and agriculturalists, understanding seed is a key to optimize germination rates, crop yields, and successful fruit production system. Moving on to our first core concept, seed is a mature fertilized ovule of a plant. It forms the bridge between generations of plants. If you look at its basic composition, a seed always contains an embryo, a food reserve, and protective covering. Think of it as a complete survival package. The embryo is the baby plant waiting to grow. The food reserve supplies all the necessary energy before the plant can produce its own food through the process photosynthesis, and the protective covering or seed coat shields the delicate interior from physical damage and environmental stress. Now, let's examine the internal anatomy by comparing the two major classes of seeds found in the crop science. It is monocot seeds and dicot seeds. On the left side, we have a cross-section of a monocot seed, such as a corn kernel. Monocots are characterized by having a single cotyledon. In this structure, the primary nutrient storage tissue is the endosperm, which takes up the majority of the space to feed the embryo. You can also see a unique protective shield called the coleorrhiza, which covers and protects the young emerging root. On the right side, we see a dicot seed like a common bean. Dicot means it has two cotyledons which split neatly into two halves. In many dicot crops, the cotyledon absorb the endosperm early on, becoming the thick, fleshy food stores themselves. Let's focus on the center label showing what they have in common. Both contain a fusiform embryo consisting of a plumule which develops into the first leaf, an epicotyl and hypocotyl for stem development, a radicle which forms the primary root, and a seed coat for protection. We also see the micropyle, a tiny pore in the seed coat that plays a vital role by allowing water absorption to kickstart the germination. To solidify our understanding of this anatomical structure, let's look at the summary on slide 4, which divides seeds part into three main parts and other supporting parts. The three core components you must remember are: the firstly is the embryo. It's the living structure that develops into the mature plant. Second part is the endosperm, the high energy food storage tissue, and lastly is the seed coat, the outer protective layer. Under the other parts, we find the specific organs that make up the embryo assembly line. The cotyledon handles nutrient processing. The epicotyl is the embryonic shoot region above the cotyledons, while the hypocotyl is the stem region located below them. At the very base is the radicle, which points downward to establish the root system, and the plumule, which points upward to initiate leaf growth. Together, this architecture gives the tiny seeds perfect directional awareness when it starts to grow. Let's look at the core topic of our chapter, seed development. As shown in the slide, using a bean plant as our biological model, Seed development occurs sequentially after fertilization and results in a mature, viable seed. In the phase 1 is fertilization and embryo formation. Once pollination takes place on the flower, fertilization occurs with the ovule initiating cell division to form the early embryo. In phase 2 is a seed growth and food storage. The young seed rapidly expands inside a green pot. The medicinal plant pump stretches lipids and proteins into the seed, filling up the storage tissue. In phase 3 is a seed maturation and drying. The seed undergoes and controls dehydration. It's losing massive percentage of its water content. Its metabolized slow down dramatically to enter a dormant state. In the phase 4, it's the mature seeds. The pod dries completely and contain hard, stable, viable seeds that are ready for disposal, storage, or planting. This is a linear chart on the slide 6, provides a clean summary of the stage we just observed. It highlights the continuous life cycle timeline, starting from the flower and fertilization, progressing to a young seed within a green pot, moving into the developing seed stage, focus on food storage, maturing into a dry seed, and finally culminating in germination. Germination is where the cycle renews. Let's analyze the two distinct cycle ways to agriculture crops emerge from the soil during the process germination. The first type we will examine is the epigeal germination, highly characteristic of bean crops. The term of epigeal means above the earth in the mechanism. The seed structure are pushed above the ground level during the growth. Let's walk through the step label 1 through in the slide. In steps 1 and 2, the seed immeables water through its micropyle, swells, cracks, open, and the radicle emerge downward to anchor the plant and drink water. In the step 3, look at the crucial moving part. The hypothetical elongates rapidly and forms a protective curved hook. This hook breaks through the soil first, pulling the delicate type behind it to avoid friction damage from the soil particles. By step 4 and 5, the hook straightens out in response to light, lifting the cotyledon entirely above the soil surface. The cotyledon expands, turn green to temporarily photosynthesize, and the first true leaves emerge from the epicotyl above them. The second type is hypogeal germination, which we commonly study in pea crops of corn. Hypogeal translates to below the earth, meaning the nutrient storage remains underground. If we observe the timeline on the picture, step 1 and step 2 look identical to epigeal germination. The seed absorbs water and the radicle grows down to form the root system. However, look closely at steps 3 and 4. Instead of the hypocortical growing, it is the apocortical that rapidly elongates because the growth happens above the cotyledons. The cotyledons remain completely buried underground within the seed coat. By step 5, only the epicotyl should break through the soil surface to develop the first three leaves. This is an incredible adaptive evolutionary strategy because keeping the food reserve underground protects it from surface predator, frost, or weather damage while the seedling establishes itself. So, we have covered the essential process of governing in chapter 1.4. It's a seed development. We explore how a seed acts as a mature fertilized ovule packaged with everything it needs to sustain the life. We compare the structure of two components, it's a monocots and dicots, and look at the timeline of seed formation and detail the functional differences between epigeal and hypogeal germination. So, as a student of crop science, Mastering this foundational system allows us to better manage planting depths, seed storage condition, and the file crop emergence. That brings my presentation to end. Thank you very much to my lecturer because spend the time and attention to watch this video. So I finish my video with Assalamualaikum warahmatullahi wabarakatuh.